Mitosis and meiosis, right? Yes. So we discussed mitosis and we discussed meiosis. Then we also discussed about chromosome structure. Chromosome structure. In this, what is the normal structure? We have a T-arm, Q-arm, or short arm, long arm, and we have centromere. Centromere. Then the terminal portions of chromosome is called as telomere. Then we discussed about the chromosomal replication or the division in here. So in the chromosome, I think this work for division, we will get a duplicate of each chromosome. This duplicate is called as sister chromosome. Sister chromatids. And I discussed one more thing. In case of sister chromatids, the centromere is called as kinetophore. Right? Then what are the basic phases of mitosis and meiosis? Yeah, P-mat. P-mat and C-cytokinases, right? So in prophase, what happens? Chromosomes will double 92 plus what is happening here? Nuclear membrane will disintegrate. Then in metaphase, they will lie in the center. And polar bodies will be polar bodies plus spindle fibers. Yeah, spindle fibers events happen there. Then here, the polar bodies, they will lie in the center like this. Chromosomes will be in the center. When in anaphase, what happens? They will be migrate towards the poles, right? The spindle fibers will take the chromosomes to the poles, right? So after in telophase, what happens?
Proper division.
Yeah. So they will be splitted and they will get their own nuclear membrane, right? Nuclear membrane will be there in the cell. Then in cytokinesis, the cell will start splitting. So splitting happens in the cytokinesis. So these are the 5 phases of mitosis or meiosis. Whereas in mitosis, first it will go for 92, then it will be split to 46, 46. In meiosis, what happens? First, 92, it will go for meiosis 1, 46, 46, this is meiosis 1, and in meiosis 2, this will occur in 23, 23, 23, 23. This is in a nutshell. And we discussed about the cell cycle also. In the cell cycle, it has G phase and M phase. In G1, we have 3 phases. What are those? G1, the S phase, and G2 phase. Then here we have mito and meiosis. So this is what we discussed. Then we also focused on the non-disjunction. Non-disjunction. What is the concept of non-disjunction?
One extra.
When the splitting of chromosomes is not in the correct number, that is called as non-disjunction, right? So non-disjunction leads to abnormal chromosomal numbers, right? So these are the abnormal chromosomal abnormalities. Okay. So first write down the names, then I will show you what are those. Anyway, the counting of chromosomes is called as chromosome counting is called as karyotyping. Very good. So usually what we will do, we will take the cells, we will culture them, then we will arrest the cell division. In what phase we are arresting? We are arresting in metaphase. So to arrest in the metaphase, we are adding a chemical. What is that? Colchicine, C-O-L-C-H-I-C-I-N, colchicine, keep in mind. Colchicine is a chemical, or keep in mind, colchicine is very poisonous, very toxic, because it is arresting the cell division. Okay, so we are using colchicine, and colchicine will arrest the cell division in metaphase. So this is the idea for counting chromosomes, right? This is called as karyotyping. Now, the normal count of chromosomes is called as Normal chromosomal count is called as euploidy. Very good. Euploidy. Abnormal is called as aneuploidy. Very good. Aneuploidy. Then now we have trisomy. Trisomy means one extra chromosome is called as a trisomy. Monosomy means one less chromosome. One less chromosome is called as monosomy. And we also have tetraploidy, etc., but they are in the plants, not in the humans. Yeah. Then, so broad chromosomes are divided into autosomes and allosomes. Allosomes and sex chromosomes, right? So sex chromosomes X and Y, and other 44 are autosomes, right? Yeah. Then let me show you a few abnormalities. Just write down these names, then I will discuss the further things. Maybe I will write here. So if there is an extra 21 chromosome, okay, this is called as Down syndrome. Extra 21 chromosome is called as Down syndrome. If there is an extra 18 chromosomes, when I say extra, I mean trisomy. Okay, trisomy of 18 leads to Edwards syndrome. Then trisomy of 13 leads to Pathophysiology syndrome. Write down all these things. Then trisomy of X leads to monosomy. Sorry. Monosomy means only one X present, another X is not present. So this is called as Turner syndrome. This is in the females, keep in mind. Turner syndrome. Then we have Klinefelter. So in case of Klinefelter, there is a trisomy of XY in the males. This is called as Klinefelter syndrome. Klinefelter Klinefelter syndrome. Then we have one more thing, that is a triple X syndrome, where there is a trisomy of X. Trisomy of X leads to triple X syndrome. Okay, and the XYY syndrome. This is called Jacob syndrome. XYY syndrome is called as Jacob syndrome. JACOBS. Okay, Jacob syndrome. Clear? Jacob syndrome. So what are these syndromes? What are the problems associated with these syndromes? That I will discuss now. Okay. So things will be somewhat, you should see the patients, then you will get an idea what are these problems are. Let me keep this cam somewhat back. See, so let us see the various abnormalities. Yes, so this is the first one. So what is Down syndrome? So this is a trisomy of chromosome 21. So this is a trisomy Down syndrome patient. So in this patient they will have mentally retarded, okay, and they have these features: small head and small ears. Then if you go for their palate, upper palate, there will be a cleft. There will be a hole in the upper palate. We have one upper palate, but there is not some hole. So we need to cross check here. And even in the eyes, there will have granulations. Granulations in the eye. So these are the facial features and problems with the Down syndrome. Okay. So this is all about Down syndrome. So this is happened because of trisomy of 20. Chromosome 21. Next one. Next one was, what is the next disorder? Edwards syndrome. So in Edwards syndrome, it is a trisomy of 18. So let us see what is Edwards syndrome. So this is Edwards syndrome, where the limbs will be twisted, unusual head to body ratio, and the ears are malformed ears. Okay. So these are the features of Edwards syndrome, trisomy of 18. So hands are fractured into fists. Okay, see, they become like fishes actually. Hands. Usually these babies, they won't survive past maybe 1 year. stop. So they will die soon. But this is what Edwards syndrome is. Okay. So this happened because of chromosome count abnormality. Keep in mind, because of non-disjunction. So leads to these issues. This is Edwards syndrome, trisomy 18. Next one. So Edwards syndrome, I want to show few more things. Anyway, this is Pathology syndrome, trisomy 13. So trisomy 13 will have this is Pathology syndrome, PATI, Pathology syndrome. So in this, the baby will have a cyclops, only single eye. Yeah, looking very, scary. The ears are clubbed. So these are few features of Pathology syndrome. Usually they will have cleft lip. Okay, cleft lip. Lip will be cutted here. See, cleft lip can be seen. And mono eye or a single eye might be present, or my eyes might be completely closed. These are very hard, severe expressions of Pathology syndrome. Sometimes baby looks normal also. Sometimes baby might look normal, but still they will have, internal organ problems. Okay. So let me go to the next slide. So these are the features of Pathology syndrome. There will be heart, kidney, and CNS malformities, and polydactyly means multiple fingers. Multiple fingers is called as polydactyly. I don't know how to pronounce. And 80% will die within the first year, and it can be diagnosed with dyslexia. Can be, diagnosed means just identifying, but not treatment. Keep in mind. We might detect it, but there is no this one. There is a test called alpha-fetoprotein. Okay, alpha-fetoprotein. This will be done after 3 months after pregnancy, first trimester. If there is any abnormality in this test, we can go for abortion. So, see, because we are living in a time where everything is toxic, all our food, everything has toxins. So we can't trust the lifestyle. All because of this lifestyle abnormalities, the cells are under stress, and the stressed cells leading to these non-disjunction issues. So whenever going for pregnancy, please, especially medical students, right? We should cross-check all these diagnoses before going for successful delivery. Yeah. At least we know that, so we need to be cautious.
So this is specifically done for only this
see, there is an alpha-fetoprotein. Actually, these are the markers which are present inside the developing fetus. Usually, they won't come into the circulation. When they will be entered into circulation, when there is a damage to an ongoing or developing fetus. Once the fetus got damaged, it has some issue. Now the alpha-fetoprotein, which is present inside the fetus, will start leaking. Leaking is not right word. It will start escaping into the bloodstream. Now we can detect this marker in the maternal circulation also. No need to take fetal from amniocentesis. Either you can go with the amniocentesis means amniotic fluid. Okay. Taking little bit of amniotic fluid, or you can also check in the maternal circulation, normal mother's blood. Yes. Usually they should not present in circulation. If they are present, definitely there is from CNS abnormality. CNS not developing properly. So there is a test, I forgot the full name, but the test is called as AAA test. In AAA test, we will detect the alpha-fetoprotein, and there are 2 more. I forgot. You can cross-check in online. AAA test. We will check this AAA test actually. Okay. If AAA got any problem, we will go for abortion. We will cross-check with ultrasound and we will proceed for abortion. Actually, abortion is not, we should not do abortion after 3 months. It is only legally we can abort before 3 months. But if these tests came, positive, there is a period up to 5th month, 5th to 6th month in extreme scenarios. Up to that, we can go for abortion only in these scenarios, not in normal. In normal, less than 3 months. That you have any to assess before going. Anyway, so Plateau syndrome. The next one was Turner syndrome. So what is Turner syndrome? In this, one X, usually females we should have 2 X, right? But in this case, they will have only 1 X, okay? Monosomy, another X has been nullified. So monosomy can be seen. So this is called as Turner syndrome. So in this Turner syndrome, the women will have short stature and their bones will be very fragile. They will have an underdeveloped heart abnormalities. And yeah, these are the few features of Turner syndrome. Okay, so folded skin, constricted aorta, and poor breast development, elbow deformities, and rudimentary ovaries. Obviously, keep in mind, almost all chromosomal abnormalities, they are usually sterile, they are infantile. Only in some rare chances we have successful birth, but usually they will be sterile. Even in the case of Turner syndrome, their ovaries are underdeveloped. So this is all about Turner syndrome. So in this, there is a monosomy of 1 X chromosome. That is called Turner syndrome. The next one, Klinefelter syndrome. So in Klinefelter syndrome, what happens? The male will have 1 extra X, right? XXYA genome. So Klinefelter syndrome is seen in males. So this is Klinefelter syndrome. So again in Klinefelter syndrome, the males will start showing the dominancy of female characteristics. So they will develop breast, their penis will be short, and there is a reduced muscle tone. So males becoming females because they have 1 active X chromosome, leading to development of female characteristics in a male. So this is all about Klinefelter syndrome. The next one was triple X syndrome. This is seen in the females, that is trisomy of X. So triple X syndrome, trisomy of X. So this is a triple X syndrome. The children looks normal, but they will have delayed motor development, low IQ. They have delayed speech, ADHD, prone to ADHD, and abdominal pain can be seen among women, among children. So this is called triple X syndrome. So where there is a trisomy of 1 extra chromosome. Okay, so both X chromosomes are active here. In this case, we will get 2 bar bodies in the females. Usually we will see only 1 bar body in the female, right? But in this scenario, we will see 2 bar bodies because of presence of 2 more extra chromosomes, X chromosomes, leading to presence of 2 more bar bodies in the cell. So this is about triple X syndrome. The next one, XYY syndrome, that is Jacobs syndrome. So let us see what is this Jacobs syndrome. So in Jacobs syndrome, what happens? The males are super males now. They have 2 XX chromosomes, so they will, they will exhibit more aggression, more acne, and obviously their height will be very tall. Okay, so they are the Jacobs syndrome. Not all persons are Jacobs. This is different story. Okay, yeah. Some things are genetic, but this is a problematic condition. So superior muscle strength and reduced muscle coordination can be seen in Jacobs syndrome. So these are few chromosomal abnormalities. So all these chromosomal abnormalities can be detected by triple X test or this karyotyping in a nutshell, right? So any gene abnormalities can be detected by 4 types of tests. Keep in mind. Genetic abnormalities can be detected by 4 types of tests. First one, karyotyping. Second one, for example, in karyotyping they are checking chromosome count. That is okay. What if there is some gene problem? How to detect a problematic gene? in a living cell. How to detect a problematic gene in a living cell? For that, usually we are doing this PCR, but PCR has an issue. PCR always damages the cell. It will damage the DNA, it will extract the DNA. It is doing, in vitro studies actually. I hope you know what is in vitro. Outside the cell test, we cannot see a living cell. We cannot observe what gene expressed, what gene activated. That is not possible with PCR. PCR will destroy the DNA actually. Sunlighting is destroying. That is the first step. 92 degrees centigrade is denaturing the DNA. So that's not possible. So the another test for gene detection was FISH. Fluorescent in situ hybridization. Fluorescent in situ hybridization. In situ means inside the cell. Fluorescent in situ hybridization. Fluorescent in situ hybridization. Okay. Fluorescent in situ hybridization. That is called FISH. The next one. The next one was flow cytometry. Flow cytometry. Next one was flow cytometry. Flow cytometry. And the final one was, even I need to study, I don't know this much. That is, one second. CGH. I don't know what is this CGH. Maybe I will let you know later. Just right now, the other test was something, cytogenetic, I don't know. CGH. I will let you know later. Okay, just write it down. So these are the 5 methods to check the genetic abnormalities. And now we have this recent one, but still it's under development, that is always this one. Yeah. So these are the methods for detection of gene abnormalities. We have karyotyping for chromosomal, gene expression FISH, and chromosomal count and expression can be studied with flow cytometry. Flow cytometry is also very interesting concept. I will discuss later. And I don't know this, I will check later. CGH and CRISPR. You already know what is CRISPR. Then, so let us see the protocol of doing a karyotyping. What is the procedure for karyotyping? So this is the procedure. So, see, this is non-disjunction. You already know what is this non-disjunction. Proper disjunction, non-disjunction. Non-disjunction leads to breakage of one sister chromatid and migrating towards either one of the poles. So here it became a monosomy condition, here it became an isomy condition. Right? Non-disjunction leads to abnormal chromosomal count. Oh, here we have one more problems. Karyotyping syndrome is very common in India also. These children, Karyotyping syndrome, loss of chromosome segment. Some of the chromosomes will be lost. The count will be there. 46 chromosomes will be there, but there will be a loss of deletion. Okay. Karyotyping. In these children, they will have cleft plate issue. Their lip will be cut here. So those children are karyotyping. Chances that there might be karyotyping, and they will sometimes you will right. Just born baby has a hole in the heart. Just born baby has a hole in the heart, right? They will do bypass surgery. Yes. Most of the times they might be karyotyping children. So they have some genetic problem, but it can be rectified. Intervention is there, surgical intervention there, but their life expectancy compared to normals, it's somewhat less. And also, some of other problems include, we just stated about the count of chromosomes, but we have other problems like inversion, deletion, duplication, and translocation. Even these things leads to problems. So I didn't discuss this much because you can study yourself. So these are few problems. I will show chromosomal abnormalities. So this is a normal chromosome, you already know. Then population also they have various names: pinocentric, aquacentric, submetacentric, and metacentric. See, why this is important? Because each chromosome number has its own location of the centromere. For example, chromosome 9 might be pinocentric, chromosome 12 might be submetacentric. So not all 46 chromosomes are same. Their location of centromere is different. Centromere was located in various portions of the chromosome. Just keep in mind. Okay. So here I given some numbers: metacentric chromosome 1, submeta 4, acro 13 and kilo. Seen mice. Okay, just keep in mind. Then this chromosomal abnormalities, count abnormality below, and deletion, inversion, translocation, duplication. Sometimes a portion of the chromosome might be deleted. Sometimes the portion of the chromosome might be inversed. Here. Okay. So inversion, that leads to some problems. Translocation leads to some problems. This chromosomes content here, this here, and duplication. So do one small work to me, by afternoon, give me examples of these four. What diseases we will see due to deletion? What diseases? Examples for inversion, examples for translocation, examples of duplication. By afternoon, you should give me examples of these four problems. Okay? Chromosomal structural abnormalities. This is for structural abnormalities. In chromosomal structural abnormalities, you should give me 4 examples.
ये तो म्यूटेशन है, ये तो अपने से चेंज भी कर सकते हैं, like जीन ऑफ इंटरेस्ट आते हैं कि किस पोजीशन का
there are some well-known diseases attributed to. We don't know specific thing, but we have on broad surface we have diseases under these domains. Deletion of a part, a segment of DNA. There are few diseases that lead to deletion, few diseases leads to. But exact gene, that is research. I agree with that point. Yeah. So there are examples you can give me later. Okay. So see here, I already said deletion, try to chart. Hemophilia is a inversion. So we already got examples. I need more examples than this. Okay. Give me more examples. I have prepared everything for you. Anyway, Charcot-Marie-Tooth syndrome, these are the things, okay? Do you brought your laptops today, anyone? You have? I mean, can you take a seminar in the afternoon session yourself about these problems? Okay, I will plan tomorrow then. I will let you know it. Let's finish syllabus first. Let's see later. Anyway, so yeah, I got it. CGH. CGH stands for Comparative Genomic Hybridization. Comparative Genomic Hybridization. I don't know much about this one, we will see later, okay? I need to study about it. Comparative, what is this? Comparative Genomic Hybridization. So, comparative genomic hybridization, we will see. So, this class is still 10:20, right?
Something about measurement.
Yeah, so they are saying that detects chromosomal copy number variation across the genome. Okay, chromosomal copy number variation. Maybe duplications, something it will detect. Sometimes I was wondering where we fit in molecular biology. When I said we, the medical and technology, do we need to go back to the genomic level? That is biotics issue. So we should only focus on the testing protocols, how to do the test, how to obtain a sample, how to run the test. So that is not our primary concern. So let us stick to it, and we need not bother much about the in-depth genetics because we are not going to do any hybridization techniques. Anyway, so applications of chromosome analysis, we already know: medical diagnosis, cancer diagnosis, early evolutionary studies, and prenatal diagnosis. This is one important thing. Prenatal means before giving delivery. Okay. During first trimester, we can amniocentesis. I say right? Amniocentesis is any idea? Obtaining a sample of amniotic fluid. Amniotic fluid sample collection is called as amniocentesis. So we will just collect 2 to 3 ml of amniotic fluid, and we can detect all types of these genetic abnormalities of our growing fetus. Okay. So yeah, they have a good chance of detection, high rate of detection. So this is it. Then karyotyping. Already you know what is karyotyping. So the basic definition of karyotyping. The definition. So sex chromosomes are specific. Why it is only karyotype sex chromosomes here? Let me see. Okay, sex chromosomes and human karyotypes. So this is about sex chromosomes. Okay, so we have X and Y. You need not to much. So bar syndrome. Bar body is seen in females, whereas bar body not present in males. Right? The concept of inactivation of X chromosome said by one scientist beyond hypothesis. Okay. Yeah. Then we discussed about sex chromosome abnormalities in this table already. So we have monosomy of X leads to Turner syndrome, triple X syndrome, and trisomy of X in males leads to Klinefelter syndrome. That's it. So these are few sex chromosomal abnormalities or their count abnormalities. Yeah. Next one. And we saw this table, we already know all types of abnormalities, various chromosomal count abnormalities, right? So a simple summary of various things. XO. So they are writing monosomy as a O. XO means Turner syndrome. Absence of one particular chromosome. Oh, see, in Klinefelter we have one more phenotype. That means XX we have double X double Y. Actually, X triple Y, we see right? X double Y. X double Y was Jacobs. Here we have double X and double Y. So there is Klinefelter can be also seen with genotype, that is double X double Y. Just keep in mind. Yeah. So yeah, these are the pictures of various chromosomes. So normal chromosome X and Y. Klinefelter we have two X chromosomes. In X double Y Jacobs we have two Y chromosomes and rearrangements. Rearrangement we cannot detect by karyotyping. For rearrangement we need to go for chromosomal staining that we will discuss. Okay. In fish or CGH that we need to go. And yeah, these are the few things. What they are saying here? Turner syndrome. Turner means XO, right? XO is Turner, right? XO. Then monosomy in females. Then why they say X only X and O or X and deleted? What is this one? O. Okay, so the second sister chromatid or second X was partially deleted. Okay, deleted in the P arm, deleted in the Q arm, deleted in both the regions. So these are the genotypes. Ah yes.
So in case of good disorders, we decompose something.
No over platform. That's why prenatal diagnosis is very important. In the first 3 months of pregnancy, just even if it costing 10,000. You know, that is okay to invest rather than having these issues, right? You can invest 1 lakh also for all these tests, even with the cost, keep in mind. Because of our lifestyle, we can't predict. Yesterday, this Dhruv Rathi, I think he uploaded one video about passive smoking issues. Rather than active smoker, you can filter and they are getting on good smokers, but the passive smokers are more affected, and passive smokers leads to many issues. I will sometimes facing this. Not just telling. Anyway, chromosomal abnormalities. We saw it. So broadly, class listen. So broadly, chromosomal abnormalities are of 3 types. See here. Structural abnormalities. Autosomal, that means autosomal abnormalities and sex chromosome abnormalities. Sex chromosome we know, autosomal we know, and structural abnormalities we have seen. Deletion, rechromosome, duplication, inversion, right? So that's it. These are structural abnormalities. So just like this table everyone, this is a summary of all chromosomal issues. So we have structural issues, we have count issues, In the count, we have segmented them to autosomal and sex chromosome. Right? Next. So broadly, these are the three things. Philadelphia chromosome, I didn't discuss with you, but Philadelphia also has a significance. It's very common. Just write down what is Philadelphia chromosome. We'll see later. Philadelphia chromosome.
Sir, in sex chromosome, the last second one.
Right, testicular feminization syndrome. That you are saying?
So, क्या है?
I don't know. We need to check. Testicular feminization. There is one interesting hypothesis I saw 2 to 3 years ago, and I will let you know. See, there is this famous misassumption about males and females, right? Have you any time thought actually the body's morphology is always a female's body? You know that fact? We are always made as a female. Then what happens? In case of males, there is a specific gene that is just suppressing X activity. Okay, this is Y chromosome actually. Okay, Y chromosome will be activated in the later stage of the development. In the last stage of development, Y chromosome will be developed. That's why all X will be the dominant phenotype. That's why, in case a male carrying one X, the X will be dominant and it will show its dominancy. The transgender characteristics, because X is very powerful. Now I just want to tell you, it's just 2 minutes video I saw somewhere animation. See, body architecture made like a woman first. Even boys are women. Then we develop some of the female characteristics are suppressed. That's why boys still have nipples. The reason was, we developed because it is a development of females architecture, but now the breast development has been suppressed. So that is one reason. Second reason, see, very interesting. So this is the uterus and this is the fallopian tube, and suspensory ligament and ovary, suspensory ligament and actually from right? Then from urethra. What they said was, see, if you see the uterine cavity, this labia majora, labia minora, they extended down. Now they have stricture in the center, and these testes started migrating into the strictured sac, and this urethra prolongated. Even the male genitalia is an extension of female organs actually. That's why in the scrotum there might be stitches, rugate we will say. So the male architecture is just a byproduct of females. So always females first in evolution. But due to cultural dominancy by humans, we give an attribution to males, but it is the nature is always a female. Mother nature. Nature always a mother, not a father. Yeah. In the beginning, there is no concept of father or mother. In the beginning, it's a conjunction. Prokaryotes, they just have plus or minus. There are few animals even today. They can be able to fertilize without the involvement of males. You know that? Some lizards, some reptiles. And the gender of the babies will be dependent on the temperature of the eggs, turtles. If temperature is slightly high, they will become males. If temperature is slightly low, they will become females. So, yeah, that is a misassumption. Only males, let there be light, let there be God, and God always created male first. That's not possible. Yeah. He should make women first. Yeah. Yes, such a shame. They say the females came from males' rib. Not at all possible. That's a different story. Anyway, I'm just telling there are some, this biblical or culturally, we have a very wrong assumption of genetics or our gender. In almost all religions, the males has been shown as dominant, not females. You see.
What's this scientifically proved?
Geez, 4.5 billion years. Yeah, everything is proved. Scientifically speaking. Yeah. In the afternoon we'll see how to do this karyotype. Simple process of karyotype. Okay, don't go to controversy, just have there is since I'm going to these issues. But these are scientific prompts. I'm not speaking some rubbish things. Anyway, thank you very much for today's attention.
Adela?